Hi everybody, my name is Jonas. I'm from the same lab as Mikan, and we're doing spatial transcriptomics. I will mostly be talking about two publications, the data for which will be olfactory bulb in the brain, but there is also some prostate cancer where you have this extremely colorful, you could call it artist's impression. No, it's really actual data, but I won't be returning to this, to this particular section. However, since we're here right now, I want to point out that this bluish region up here is what our transcriptomics data picked up as some kind of interesting region. And according to the pathologist, a smaller region to the center of this is a solid tumor, indicating that our technology picked up this larger region, including a disconnected region over here. So that was fairly interesting. The paper I will mostly be talking about is out on the bioRxiv, and you could fetch it using this QR barcode. Anyway, so I will probably be quick with the introduction of the ST technology, since we just heard about that. And we'll proceed to address two little points in the visualization of spatially irregularly placed data, and how generally to visualize such spatial information. And we'll then be coming to talk about several analytical approaches, focusing on Poisson regression, and then in a logical manner, so to say, proceed to negative binomial regression. I'll have some results, and if there's a little bit of time, I might be talking about future directions. So here we go. I think we covered most of this, and I perhaps only want to stress that we have, if you want, mini bulk samples within the spots, since we get about 10 to 50 genes load of mRNA on a spot. And it perhaps was not quite perceived earlier, but there are these UMIs in here, which got introduced earlier in the morning. So we don't really have an issue with PCR amplification artifacts. The staining, as was remarked, is hematoxylin and eosin staining, the standard for pathology. And I'm not sure if not perhaps fluorescent-based microscopy using antibody labeling might be interfering a little bit with the process. This is not really clear, but what we have now is definitely what has been used for decades in pathology. Yeah, I'll skip this. This was explained just now. Basically, we will only be talking about what to do with the data once you have it in the form of a count matrix with genes and spots. So the first point I want to make is that due to the printing process introducing slight geometric inaccuracies, including, for example, missing data points and the points not being arranged in a exact grid, I think it is important that we spend a little time to use the right visualization for that. Because, for example, if you were to choose point size to indicate expression strength, or you use a simple single channel palette where the background color is identical to low expression, then, for example, missing data will not be obvious or will not be distinguishable from low expression that was actually measured. For this reason, I think it's good if we can maintain such kinds of matrix plots, which you can easily make in the case of regular spatial data in the form of an exact grid. And this we can do by using the Voronoi tessellation, which both accurately maintains the spatial information, as well as is informative of places where we are missing data. Of course, we could go on to use probabilistic models to properly interpolate between the data points, but just for visualization purposes, I suggest people keep the Voronoi tessellation in mind. One of the first things I ended up doing when I joined the lab was to apply dimensionality reduction techniques in order to directly visualize information. And the idea is fairly simple, really. You have a matrix with the spots in the rows and the genes in the columns, and you simply apply dimensionality reduction to that so as to reduce the number of columns to 3, which you then, after squishing into the unit cube, basically use as coordinates in color space, so that you can assign 3 color channel bits and combine that in one colorful plot, somewhat maintaining, hopefully, most of the information of the original input matrix. The obvious choices for these dimensionality reduction techniques are PCA, or TSNE, or UMAP, for example, but the choice is yours. This works rather well, actually, as I would like to demonstrate here with one section from breast cancer, which on the right side shows the bright field image that was obtained in the process, annotated by a pathologist using different colors for different anatomical features. Such as, for example, there's glands that are healthy in green, there is fatty tissue in blue, there is a solid tumor in orange, and there's invasive tumor cells as well as immune cells in yellow here. You will observe that if you do this dimensionality reduction-based visualization of the information that we get from the transcriptome, we get a rather neat mapping between that. For example, what's green here is also in this mint shade of green here, and the fatty tissue is mostly in this yellowish green. That's fairly concordant. You will also note that between the invasive tumor and the solid tumor, we have purple versus pink. I do want to go on to note that if you have several sections from one experiment, this can still work rather well. There's interesting things to be seen here, like this tumor focus in the center not being present in the 4th section anymore, as it should. If you do go on to look at more, yes.
So when you analyze this data from just this, so do you have like batch effects, like that you have, that each slide will have, there will be some biases towards, I don't know. Also, even inside in each area, do you have like around that's often the case if you do an experiment, that somehow on the borders there is some different effect, or between each slice and so on?
Yeah, that's a good question. The batch effects is exactly what I wanted to get to now. As for the other point, I guess you're referring to positional effects on the arrays. Yes, such effects might be present. They would be more pronounced, presumably, towards the corners. And the tissue is typically placed towards the center. And we only consider those places, really. We haven't studied such positional effects sufficiently yet, but you could take subsequent sections and put them on multiple arrays in different orientations to address that.
Sorry, this picture before, it was from only one slide?
No, these are 4 sections, each separate.
I mean, but one picture.
Yes.
Is it from one slide only?
Yes.
Although a lot of information is lost on the when you do the single cell process.
This is no single cell approach.
Also,
this is
no advanced transcription here. Because before it was said that during reverse transcription we lose like 80 to 90% of the data.
Yeah, that's right.
And so there's no reverse transcription here?
Of course, there's reverse transcription involved as, I think, one of the first steps in the library preparation. In fact, I would like to correct the efficiency estimate that Mikan had for our technology. I would put it a factor of 10 lower at 0.5% overall efficiency. My calculation is due to having about 5,000 or let's say 10,000 molecules in a spot for 50 cells. If you put 100,000 molecules per cell, that's how it comes down. But yeah, but again, this is no single cell sequencing technology. Yeah, so I wanted to come to speak about batch effects, and they become obvious as you look at multiple samples. In this case, we have 16 olfactory bulb samples from, I think, about 7 individuals, and I can point out that the first individual had 4 sections. The second had 1, the third had 2, the fourth has these 2, and there's a triple, and this is 1, and finally this is another one. I can also say that the last 4 experiments were done months after the first one. So you might be thinking, well, that's the different day of experimentation. However, if you leave those experiments out here, the last 4, then you get batch effects visible between some of the other samples versus the others. This is in line with the TSNE highlighting the main points of variation and somewhat neglecting the others. With this, I also would like to highlight that one should conceive of TSNE really as a visualization technique as opposed to an analysis technique. Yeah, and the remainder of the talk, I would like to explain how you go from this here to that, in which I hope to convince you that we will be able to successfully address these batch effects so as to achieve a consistent colorization between corresponding spots in multiple samples. I would begin by suggesting that we approach this from a mathematical angle in which, excuse me once again, in which basically we think of reducing the information literally by conceiving of the matrix as a product of 2 lower ranked matrices. Naturally, in most cases, information would be lost if you reduce the rank. We also should consider that really what we're dealing with are non-negative numbers. So we should really strive to achieve a decomposition in which also the decomposed matrices are non-negative. This would tip.
So the X is not is
that's the data.
But the rows are the spots and the columns are the genes. What's
at this point, this doesn't really matter. This is illustrative, but I will have definite indexes on the next slide. The point is that if you count the number of entries in the 2 matrices that you multiply together, they will be only a tiny fraction compared to the original input data. We could, for example, have 20,000 genes and let's say 20,000 spots, so we end up with 4 times 10 to the 8 numbers on the left side. And we might have on the right 20,000 genes and reduce it to 20 dimensions, so that's 400,000 numbers, and 20,000 spots in that's another 400. So we got like 8 times 10 to the, what is it, 5 or 6? In any case, it's like 10 to the minus 3, roughly, the compression. And but the objective function used to perform this is basically a minimization of the L2 norm of Euclidean distance between the input and the reduced matrix. And it's not really clear that a discrepancy of value X for this gene in that spot and should be the same as that discrepancy over here. So this mathematical definition is not what I want to suggest we do. Rather, we should be using a probabilistic formulation in which we use some model so as to say that the data that we see, for example, should be the expected value of, well, something that our model tells us. We could be using latent Dirichlet allocation, which is fairly frequently used in document modeling. The analogy is that the spots are the documents and the gene counts are the counts of the words in the document. And so it's a problem of inferring the unknown topic distributions in your documents. The topics now being cell types in our case. The LDA is formulated in terms of Dirichlet distributions and the multivariate. And Poisson regression, obviously in terms of the Poisson distribution. And I'll come to talk about the Poisson negative binomial more extensively later. I do want to highlight that both LDA and Poisson do not allow us separate control of the variance, and it is well accepted in the field that the variance of gene expression is larger than just shot noise. So with this, let us turn to Poisson regression, which has been described in this method, in this publication in which we applied it to prostate cancer. But I won't be having any further pictures on that. You would have to go back to the title slide, that picture that I had. So here's a little graphical model that is really at the core of Poisson regression. We assume that the observed counts are given as a sum over some hidden counts, and that these hidden counts are indexed by a cell type over which we have to sum to get the observed count. The Poisson distribution has a rate parameter lambda, which for now, for reasons that will be clear in just a moment, we assume is given as a product of some expression intensity and some weight of that cell type. You will perhaps notice that due to linearity, if x is the sum of some Poisson distributed numbers, then x itself will be Poisson distributed. And the reason that we can hope to achieve such a decomposition is because we have information sharing. That is to say, in order to infer the expression strength of this gene in that type, we can look across all of the spots. And in order to estimate the mixing weight of that type in that spot, we can look across all genes. So there's there's mutual information sharing here. Overall, again, we only observe the mixtures here, so it's a decomposition approach where we don't really know both the underlying gene expression profiles as well as how strong each type or cell type is expressed in a given spot. We can go on and perhaps overload the notation a little bit to say that the entire matrix X is Poisson distributed with this rate matrix. And thus, we basically can write that X is roughly the product of these two matrices. So with this, we have a clear probabilistic interpretation of what it means to decompose a matrix, which is more satisfying than just choosing the best matrix according to minimizing some mathematical norm of the difference to the input. I would like to mention at this point that in this publication we allowed for some covariates, some specific covariates. For example, one for each sample, which would allow to change the gene expression baseline for, in that section only.
So you directly go from the XGTS to XGS by summing up, right? And there's no place in this model where there is accounting for errors. Because you were saying the genes can be just missed, right? They are there, but we are missing them. So wouldn't this a good idea to explicitly model these events when you're because you're saying, okay, it just depends on expression profile, which makes sense, and mixing weight. But there's no...
Yeah, I mean, really I was, brushing a little bit about the details. So this, while I write it here as a product of two matrices, you can also multiply in vectors for the spots, for example, or for the genes. So in this way, you would have spot scaling factors, which would address entire rows, columns in this matrix here, so as to downregulate all of the genes expression in that spot. But generally, perhaps already this would be enough to answer your question. If some type is lowly expressed in a spot, that's here. And if all the types are lowly expressed, that would also be here.
That's a parameter that you estimate.
Yes, that's right. We didn't specify on this slide any priors for these matrices, but that is obviously necessary. And in this publication, we had mostly gamma distributions as priors here. But
and the large T, are you giving this a priori?
Yes, that is to be determined by the user of the application and will require experimentation. I mean, it's really difficult to come up with principled ways of determining such things. Ultimately, we would have to look at the likelihood of all possible models and then, compute the posterior from that. That is not feasible, really. And if we just have a series of maximum likelihood estimates for, 2 types, 3 types, 4 types, and so on, I think it's effectively better if we just leave this up to the user. They have to do a judgment call, ultimately. I mean, we can supply further information, but yeah.
Couldn't you place a prior on it, like in the sense of a Dirichlet process?
Yes, I agree. But the inference is a bit costly right now for one particular T. So in inference, we don't vary T right now.
How do you infer the parameter?
Sorry?
How do you do infer?
So this model enjoys conjugacy. So we can do Gibbs sampling, actually. That is quite convenient. In the model that I will talk about next, we unfortunately have to rely on optimization, which means it's a little more brittle in its inference. Yeah, and now I would like to extend this to negative binomial regression. This is figure 1 from our paper and supposed to give an overview of the general setup. It indicates that on the left we have 4 samples, for each of which we do spatial transcriptomics separately, so that we end up with a set of gene maps, 1 for each gene and for each sample. Now these gene maps are quite numerous. There might be 20,000 and you might have 10 samples, so you got to look at 200,000 pictures. It's a little bit much, and I hope that this approach would allow you to compress this information in an interpretable manner. Now, the actual method is this here. It's composed of first a step of inference, and then we compute expected marginal counts, which we can visualize in terms of the spot and type dimension, or the gene and the type dimension. So here we have 4 types indicated. They have these, and the spot and type dimension I would call spatial activity maps, and the genotype, let's just call them expression profiles. These are point estimates we're displaying here, of course. And for this approach, again, due to using optimization, we cannot really very well give uncertainty estimates here for these.
Can I ask a question about the previous one? Just one question. Sorry. So when you say, because of this mixing weight, I was just, so, okay, from this approach, can you, for example, say, because I used this, I estimated my parameters, I will now say there are 10 cell types, and each of them is present in this sample with this fraction, right? So 50%,
precisely.
yeah. But now you have this, and you will be getting this from the mixing weights.
Yes.
But then now they also incorporate this error, right? As you said, so they're not exactly the true abundance of the cell types.
Yeah.
They also say how much you actually had it after.
No, yeah,
That's
this
how
is
it
outside of
is.
the model. I would say that what we're modeling here is really only the number of counts that we actually end up seeing on the array. Yeah.
Yeah.
And as for how much of this type and how much for that, sure, you can look at these parameters, but since we have these hidden counts here, we can do statistics on them directly. These are kind of the decomposed counts.
Yeah. The count, that was the
Yeah.
true, the true observed.
Yeah, well.
Yeah.
So again, so we would perform inference and then compute these expected marginals to yield these activity maps and the gene expression profiles. Important ingredients and really crucial to extending the analysis to more than one sample is that we reflect the experimental design, which here is indicated to contain samples from 2 individuals. And this is just mock case control. They're really replicas in this case, but in order to illustrate the fact that we can use additional covariates, I included it in this way here. And then the inference also very much depends on the probabilistic model that you use, and that equally has to be adjusted to the application by the user. The model consists of an invariable part, that is the core model, which is very related to the setup in the Poisson regression that we had, which is that the observed count is given as a sum of some hidden counts, and that the hidden counts are distributed according to some probabilistic model. In this case, the negative binomial. Now the negative binomial takes 2 parameters, and for these 2 parameters we then allow the user to specify regression equations, logistic regression equations in this case, and the user is free to use coefficients that are indexed by nothing, for example, or by the gene in the type, or by the type, or by the type in the spot. All of these are very much related to these parameters here. But in addition to that, and crucially, the user can specify coefficients that are indexed by what is specified in the design. For example, here's a coefficient indexed by the gene and the section, or here's one for the gene and the individual, or gene and treatment. And in this way, we can make sure to allow the model enough variability so that it can fit and ultimately correct for present batch effects. Finally, the third part of the model is whatever structure you want to put on as prior on these coefficients. In this case, just standard normals. But our framework allows for arbitrary hierarchies composed of beta distributions, gamma distribution, normal distributions, as well as something I'll come to on the next slide, the last slide. I also want to note that even if we reduce the information from about 20,000 genes to, say, 20 factors, of which 4 are displayed here, it is still useful to summarize that information such that it can be perceived at a glance, using the techniques I spoke about initially. And this I explained already. I just perhaps want to highlight that the second parameter of the negative binomial, this odds parameter, and as a parenthetical remark, we're not using the Kp parameterization of the negative binomial, where P is a probability parameter. Rather, we're using the odds. This is a lot more compatible with the logistic regression that we're performing. But crucially, I want to remark that the second parameter, which is essentially determining the variance to mean ratio, that is to say the over-dispersion of the negative binomial, this parameter is not allowed to depend on the type. In other words, regardless of what the different cell types figure in terms of mean expression for a gene, the variability around that mean is in this model not dependent on the cell type. The reason for this is computational convenience. But probably more importantly, probabilistic reliability. I think we're still far from guaranteeing that all of the parameters which we infer, which number in the hundreds of thousands, actually have the values that they should have. So I'd rather err on the conservative side and estimate the mean correctly and separately for all the types, and leave the variability to be only a function of the gene. Yeah, and with this, I guess I can say, if you don't like maths, it's all good now. that was it. I want to show results. So here's on the olfactory bulb. In this analysis, I actually used 16 samples, the same 16 that I had earlier on a slide. And there's only 4 samples shown. They're in the columns here. The bright field image, or its inverted version, is up there. And the different rows then are the spatial activity maps, as we call them, for the different factors, of which here also only 13 are shown. There's another 7. We used 20 factors in this analysis. Down here you see a summarization using TSNE. Here is a summarization using UMAP. And here is a hierarchical clustering of this activity map matrix. The points that I want to make about these results is that we observe like a high degree of consistency. For example, here is enlarged the same pattern in 2 samples. And you will notice that, well, although they're rotated, they are really very similar. Furthermore, we pick up numerous samples that, how do you put this, stack, that have an inside-outside relationship. And there's relatively few factors that we need to capture the residual noise. You might, for example, notice that here number 12 doesn't have so much structure in it. And perhaps number 6 here is not completely consistent across the samples, but overall this is pretty good, I think. To go on to further illustrate this, I would like to show these brain sample results, in which we now, in fact, only have 4 samples as indicated here. And we used 20 types, all of which I display here. I took the liberty and annotated the anatomical regions that are highlighted by these patterns in this column. And as you will see, for 3 I had to put a dash because I couldn't really infer something that isn't common here. They do pick up structure that is specific but not shared across the genotypes. So this is one genotype and this is a second genotype. Sorry, individual really. But other than that, 17 of these 20 factors that we identified here have clearly corresponding anatomical regions. There is, for example, 2 that identify Ammon's horn, region 1 and 2, and the second Ammon's horn, region 3. And there is another one also within the hippocampus that is identifying the dentate gyrus. And we will be looking at the spots that correspond to these regions that we thus defined in a multi-sample analysis for differential gene expression analysis on the next slide. So here's the result of performing the differential gene expression analysis. The point here is that because we did a multi-sample analysis, we get a consistent classification of the spots in multiple samples. And for this reason, we have greater statistical power to look at smaller anatomical regions to perform differential gene expression analysis. The plot here shows the mean expression on a logarithmic axis and the fold change between the 2 regions on the vertical axis. And 2 of the most extreme genes, PROX1 and FAM163B, that are up in the dentate gyrus, are quite clearly specific to the dentate gyrus if we look at images retrieved from the own brain atlas. Whereas NeuroD6 and KHDRBS3, which are among the most extremely downregulated in dentate gyrus, versus Ammon's horn, we clearly see Ammon's horn being its main source of expression. So with this, I hope to convey that indeed, by modeling the data across many samples, we can identify the corresponding regions and then perform downstream analysis. Because everybody likes TSNE scatter plots, I had to create some as well. I'm not really 100% sure what the upshot here is. Definitely what we see is that, so notice that the colors are matched here. So this you saw before on the preceding slides, and you also saw this before on the preceding slides. And so we have here a color gradient from yellow to light to blue to green, dark blue, red, from the inside to outside. And it looks like we have a continuous manifold, or continuous sampling of the manifold here. On the other hand, for the brain sample, we have what looks like disparate clusters, but hopefully all of you know about this crucial parameter of TSNE that you want to play with, the perplexity that somewhat dictates how clustery or continuous the reduction will be. So my guess is that in this case our spatial sampling is not dense enough so as to get points in the transition so much here as we get it over here. But the more important message of this slide is that if you contrast these kind of automatic results that we get with what you have in a reference anatomy atlas, there is an extremely strong concordance. I mean, just imagine performing one experiment and getting the same result that people had been accumulating over years. Yeah, and with this, I just perhaps want to highlight that the direction of development right now is to incorporate actual spatial modeling into our system, and that would be using Gaussian processes. The trouble that we have is clearly we want different length scales for different factors, because some factors are, well, changing with the high spatial frequency, and other factors are broader. And what people typically do with Gaussian processes is that they consider a grid of these length scales and then select the best. But unfortunately, we can't quite do this because the Gaussian processes are embedded rather deeply in our system. And so this is a bit unwieldy. And I guess the way that I want to approach it right now is really as a sum of multiple Gaussian processes. And yeah, this is what we are just working with. I would also like to mention that another two directions that we want, independent of Gaussian processes now, another two directions that we want to move in is towards integrative modeling of our ST expression data with the histology that we also acquire. And the most obvious way of doing that for now would be using convolutional neural networks to, for example, predict the transcriptome from the histology, which is a bit of a pixel corruption problem. But really what we want to aim for is a generative model that gives rise to both at the same time. And finally, as a third point, it would also be to integrate single cell expression data with our ST data. And yeah, that is the final point I wanted to make. As for conclusions, well, I presented to you a probabilistic model to decompose spatial transcriptomics data, and I hope to convince you that this is helpful in figuring out what the important determinants in the data are. Yeah, and with this, I'd like to highlight that Jens, who's here, has been working with me. And we have the student Ludwig Berdienstrolle, who has been very helpful in working on this. And of course, the experimentalists are our only source of well, work. So thanks to them, of course, as well. Yeah, and with this, thanks for your attention.
Are there any questions? Yes. Yeah.
Great topic. When you have this large, basically all this, very early on, you said that you can correct the batch effect. For example, you're including, preparation date and whatever other processing batch effect into your negative binomial regression. Is that essentially how you fixed it? Because you didn't mention.
Yeah, actually, I mean, what? Let me talk about this first. We only use discrete values here. It's not like a linear regression where you say, for example, like the further into June you are, the worse it gets. These are just labels, really, that we don't interpret user-annotated covariates in a quantitative way. For this reason, these coefficients are really completely free to, subject to fit and subject to the prior.
But you mean you could have multiple covariates? For example, you could have, which date they were processed and all that.
Exactly, which experimenter, what treatment conditions and such.
Okay, and then in that case, when you go later on to look at the actual differential expression, do you simply get those, like this one, you get that from the regression model?
Yeah, actually, good that you ask, because really the DGE part here is fully reliant on DE-seq. We don't actually use our own parameters here. The way that this workflow was is we model the data and we end up with classifications for the spots, and then we select those spots and go to the original data to input that into DE-seq, providing it with the same covariate information that we have.
Is that something, because your model seems to be like almost perfectly capturing the information already.
Yeah, that's right, but we just haven't made the time to figure out the statistical test to do it. So if anybody has inputs on that and I could help, I'd be more than welcome to discuss that.
Any other questions? Okay, so if not, let's thank the speaker again.